Hello and welcome to today's webinar, 2026 Trends in Clinical Trials, Accelerating Clinical Trial Development. Today's webinar, presented by nQuery, should provide you a preview of what we expect to occur in 2026 in clinical trial design, and indeed some of things which have already come to fruition as we're coming a little bit later this year, and then also hopefully provide you context on what this means for your clinical trial design. Before we get started, just some frequently asked questions. Firstly, is this webinar being recorded? Yes, the webinar recording and slides will be sent to you after this webinar is complete later today. If you have any questions or technical issues, please use the questions option in the webinar software. If time allows, I'll answer questions at the end. Otherwise, I'll answer all questions via email after the webinar is complete. So, as I said, welcome to today's webinar, 2026 Trends in Clinical Trials. It's already been quite an exciting year in clinical trials, lots of significant changes happening in the regulatory world in particular, and hopefully give you a preview of what's occurred there. We're coming a little bit later this year. We are changing to a new webinar schedule of every 2 months. And so that's given us an opportunity to not just give an idea of what might happen in 2026, but also give you an overview of what's actually already happened in 2026 and what that might mean as we go forward into the rest of this year. Just in terms of myself, my name is Ronan Fitzpatrick. I'm the head of statistics here at nQuery. I've been with the company over a decade now. There's been huge changes in terms of adaptive design, Bayesian methodology, and many other areas during that period. I'm very excited to continue to see and evolve our software in response to that as we go forward. So in terms of today's agenda, firstly, a new era for Bayesian trials. Obviously, this new FDA Bayesian guidance that came out just a few weeks ago is a huge milestone and has significant implications going forward. Then we'll have a goal for continuous trials, this new concept coming especially from the FDA of having trials which kind of seamlessly move into each other rather than having the kind of traditional three-stage clinical trial process. And then we'll talk about some other trends for 2026 that might be important, like AI and ML, multi-regional clinical trials, rare diseases. And then, as I said, if time allows, we'll have some conclusions and discussion at the end. Questions welcome throughout, but as I said, I'll answer if I have time live later on, but I will answer all questions via email afterwards. So please do put them in if we don't get time at the end. So today's webinar, as mentioned, is brought to you by nQuery, the complete solution for optimizing clinical trial design from preclinical research to confirmatory trials. And you can see some of the options available in nQuery on this slide here. nQuery is a very comprehensive solution for clinical trial design and use a tiered product from base to expert, with base focusing on sample size and power calculations for fixed term trials, plus focusing on our Bayesian features, pro focusing on our adaptive design features, and expert focusing on simulation for milestone prediction and similar. If you want further detail on the tiers, there's a URL here in the slides, which will be sent to you after the webinar is complete. Okay, so let's provide a high-level overview of what is happening in 2026 and as we enter 2026, what kind of things have been going on. I think the biggest thing that we're seeing at the moment, especially in the FDA with new leadership in charge, is that there's been a big push and concern about the increasing costs, the long timelines, and indeed the increased global competition for clinical trials that's occurred, which is leading to the kind of traditional powerhouses such as the EU and the US kind of looking over their shoulder a little bit and asking, okay, what should we be doing to ensure that our clinical trial process is competitive with that? Now, that's an ongoing process that's been going on for over a decade at this point, and you've seen guidances and things like adaptive design from the FDA, and obviously the major milestone of ICHE 20 published last year, or at least the draft version of it, which I covered in detail in webinar last year. If you're interested, there's a URL in the references at the end of this slide deck if you're interested in that. Master protocols, platform trials were obviously a major focus during the COVID-19 pandemic, and we've just seen a massive shift in what's possible there and the number of those that have proliferated recently. Real world evidence is another area of major interest in recent years. And so what you're seeing here is that, the largest regulators, the FDA in particular, is responding to this and basically saying, okay, what should we be doing to change things to make sure that we can stay competitive and we can get treatments out to patients in a timely manner and in a manner which encourages as much therapy development as possible by reducing costs. And so I think what you're seeing here is that a lot of questions or a lot of assumptions which previously would have been considered to be sacrosanct are now being torn up and being put away. So, I think this idea of the totality of evidence, where basically you're looking not just at clinical trials, but also looking at other sources of evidence, earlier stage evidence being used more directly, and all of that feeding into accelerating approvals and improving the performance and accuracy of these trials, with maybe also relying a little bit more on, like, say, phase 4 or post-marketing surveillance powers to take back if there are mistakes made in that situation. And so there's basically been a bias towards, I would say, speed over some other concerns in response to all of these underlying trends. And then two major areas which have already basically seen like significant progress in late 2025 and early 2026 is Bayesian Statistics, which kind of feeds into a whole bunch of these other trends in terms of the inferential biostatistical side, and then a greater demand for continuous trials, especially from the FDA. And there's also been a number of other trends, like the continuing growth of AI/ML in clinical trials. Rare diseases is seeing basically a complete overhaul in many ways in recent years. Where obviously this week, if you've been paying attention, the FDA had is having its rare disease week, and some major announcements, or basically the culmination of major announcements occurring there. And then multi-regional trials maybe being the negative side of this, where if these kind of carrots don't end up working, will we start seeing some sticks come out? And so I just think this is it's a really interesting time to be involved in clinical trial design and clinical trials generally. And so I can hopefully provide you an overview of these kind of two major areas of Bayesian statistics and continuous trials, to kind of give you a flavor of what we might be looking at in the years ahead. So part one, new era for Bayesian statistics. We'll talk about what that new era is in a moment, but let's just first provide a brief overview of Bayesian statistics and Bayesian clinical trials so we know where we are. So obviously Bayesian analysis is very popular statistical methodology where we characterize a prior to use basically a distribution for what we expect the parameter to be, and then we get some actual data from, say, a clinical trial, and we use that data and the prior to get what's the posterior distribution. So based on what we believed previously and what actually happened, we now have a future updated distribution of what we think the parameter, say the mean difference or the hazard ratio, is actually going to be. And so that obviously is combined in this Bayes theorem, which you're all fairly familiar with. And this is all kind of conceptually interesting from a biostatistical point of view. And obviously there's lots of arguments about the relative merits of Bayesian statistics versus frequency statistics. Bayesians would argue that Bayesian statistics is more intuitive, in the sense that rather than conditioning on the hypothesis and then saying how likely it is given the hypothesis, the Bayesian interpretation is more how likely is the hypothesis given the data. But we don't need to get into that really right now. There's lots of other practical reasons why Bayesian statistics has got increasing popularity, especially things like its flexibility in terms of, like, say, continuous monitoring, and its ability, especially its ability to integrate previous data via the prior mechanism, rather than being a bit more rigid in the frequentist framework. And so, I think we've already seen a huge growth in Bayesian methodology, especially in early stage clinical trials. So in phase 1, we've kind of seen model and rule-based algorithms replace the kind of 3+3 paradigm. And this basically leads to a huge amount of evolution and change that's happening in the phase 1 stage. And I think you'll also see some of that following into the continuous trials concept for early stage trials that we'll talk about later on. But I think, basically there's kind of either kind of purely Bayesian approaches, such as CRM, which is basically a fully Bayesian approach where you have a skeleton dose escalation curve, and that's been updated each time you add a cohort of subjects, and then obviously that's used to select the best the best dose level to be checked to find the maximum tolerated dose, or the OBD, or whatever is whichever is your particular target or targets of choice in your phase 1 trial. But even rule-based or model-assisted methods like MTPI2 will have a large Bayesian component within them, and even ones that aren't really Bayesian, like I3+3, will tend to include things like some kind of Bayesian rule for overdose control in many cases. So Bayesian is kind of very central to the modern approach to phase 1 dose escalation trials. And then at phase 2, pretty much all types of phase 2 trial have seen some level of Bayesian adoption, whether it's kind of your more classic proof of concept Simons-like designs, where Thal and Simon and Bob 2 have found some leeway. Dose finding studies are very commonly done in the Bayesian framework. Similar to the dose escalation, you basically are able to update your dose response curve in an adaptive fashion very easily within the Bayesian framework. And then massive protocol designs, basket trials in particular, are very strongly incentivized to use Bayesian designs, where the borrowing concept, especially in basket trials, can be very useful. And so this growth, as I said, is underpinned by this additional flexibility and its ability to integrate additional sources of evidence, use the totality of evidence much more easily than in the frequentist framework. And so Bayesian methods are already used in phase 3, and we've seen implementations, particularly for borrowing or for pediatric extrapolation or similar type of concepts. And you can see the complex individual design program if you want to see some case studies of that. But the major milestone that we're going to spend a lot of time here today is this recent draft guidance published on Bayesian analysis. And this does cover all stages, but it's particularly important, I think, probably for confirmatory trials. And so this draft use of Bayesian methodology in clinical trials of drug and biologics guidance for industry published in January, so only a handful of weeks ago. And if we'd done this a few weeks ago, we would have been predicting what's in it rather than saying what's actually in this draft guidance. And it's got a very big push from the FDA, and it continues to be mentioned multiple times, including by the current FDA Commissioner, Mark Macquarie. And you see here this quote from the press release for the release of this guidance. That's a major, a sign of his interest and how much power he thinks that this new inferential approach could have for many of his other priorities that he has. And so Bayesian methodologies help address two of the biggest problems of drug development: high costs and long timelines. Providing clarity around modern statistical methods will help sponsors bring more cures and meaningful treatments to patients faster and more affordably. That's a pretty big thumbs up, I would say, in regulatory terms, and I think a real go for it attitude coming from the FDA if you were thinking about using Bayesian methodology in your clinical trial. And so what I've actually done here is basically just kind of a section by section overview of this, very short. Obviously, I'm not able to go into the weeds given the time available, but I think it's really, really useful guidance. It's very detailed, has lots of real world examples, and kind of goes step by step what you need to think about if you're going to be doing a Bayesian clinical trial. And so the early stages basically outline the key concepts, things like the prior, the posterior, the likelihood, and then actually provide a series of real examples, including some from the complexity of design pilot program that they've been running for several years at this point. And I would say there's a big focus in this guidance on borrowing. And I think in particular, when we get into stuff like the type 1 error calibration and stuff like that, basically they're kind of saying, we think that the value of borrowing, I should say, useful information, so from prior studies which are in a similar space across disease and subtypes between subgroups, that value outweighs what is basically sometimes the intrinsic type 1 error inflation expected in that scenario due to the, depending how much the prior and the actual reality disagree from each other. And so these real world examples go from borrowing. So for borrowing, you see case studies like for antibiotic studies, borrowing from previous studies. For diseases, you see specific reference to basket trials, to studies involving epilepsy. Between subgroups, you see things like multi-regional clinical trials via a hierarchical model of diabetic studies. You then see things like external and non-concurrent controls in the context of platform trials like GBM Agile, and multiple sclerosis in the context of pediatrics, for example, and other pediatric extrapolations given its own explicit section. It's given an example of type 2 diabetes. And then in dose finding, there's a big emphasis on the kind of dose escalation, early phase dose finding there as well. So, these real world examples are really useful because then you can review those studies, see the protocols, see the papers that come out of that, see the case studies written up by the FDA themselves for the CID relevant ones. And that's really useful to kind of establish a baseline, okay, of where we're going. Because I think, there's been commentary basically saying, like the early move has already happened. The early movers are already moved. We're now in the stage of like widespread adoption. If you kind of technology adoption curve, this is the point at which people get on board because the train is going type of situation. So very exciting stuff there. They give a good explanation of kind of what would be a classic Bayesian success criterion, which is mostly given in terms of when I say the posterior error framework. I think there was some commentary that I saw that suggested that the Bayesian predictive probabilities could be integrated better into this, which is like basically it's kind of like prediction interval versus confidence interval. Basically, this would be like how likely is future data, going to be X rather than just the prior, probably the parameter related inference testing. But that's kind of getting into the weeds a little bit. And basically they say explicitly that type 1 error may not be applicable or appropriate. And then they specifically mention, especially in the context of borrowing. I think the fact that they're saying there are studies where basically we think the Bayesian approach is better. That's basically what they're saying. There's nowhere that they say that the Bayesian, like the frequentist approach, is definitely better than the Bayesian approach, as a vice versa. I suppose that the assumption there that obviously because frequentist paradigm has been so common up to now, that there's going to be lots of cases where the methods are much more well established for frequentist paradigm, so there's kind of intrinsically an advantage there. But it is interesting that they go that far, basically saying it's not applicable or appropriate of using type 1 error control. And obviously, I am conflating the idea of type 1 error control with frequentists, but obviously, as they say themselves, it is possible if you're using a Bayesian methodology or some aspects that are Bayesian to have calibration to ensure that under the null hypothesis, so if we assume that there's no effect, the effect size is 0, or the hazard ratio is 1, or something similar, that basically the type 1 error calibration is possible. And that might be of interest, especially if we're talking more using Bayesian methodology, like Bayesian success criteria in the context of a complex adaptive design, or in the context of where we're using non-informative priors, where it's known that for many kind of standard non-informative priors that the Bayesian and frequentist difference will tend to be highly correlated to each other, or highly similar to each other. And basically, just to say this, like P, D greater than A greater than C. So A would be your hypothesis. So D is your parameter or whatever of interest. But A would usually be, so for example, for a superiority trial, that'd usually be 0. For a non-inferiority, that might be like -1, your non-inferiority margin, and stuff like that. And basically, the C would be your probability of success. And so, for example, for type 1 error calibration, that might be set to well, this is 2.5%. I should be saying 97.5%. But basically the idea is that you can calibrate under the null that to get the type 1 error calibration if you're interested. But basically, you're usually targeting some level of success criterion. So if you're using a pure Bayesian approach, then you're usually going to be making inferences based on the posterior error itself. So they give an actual explicit example of the probability of D greater than A. So imagine the probability of mean difference is greater than 0. The parameter specifying the mean difference is greater than 0 is greater than 98%. So you're saying that there's 98% chance that the mean difference is greater than 0. And that might be your success criterion here. Now, I don't want to get ahead of myself, but one of the very interesting things, I'll talk about this later on, is, okay, what will be the emergent standards, emergent precedents set when defining, clinical trial thresholds when conversations occur between sponsors and regulators in this scenario. Well, I definitely know that what people don't want to happen is that 98% probability for P theta greater than 0 is greater than 98% or 0.98. That's now the equivalent of the 0.025 alpha criterion. I imagine that is exactly what Bayesians want to avoid in that kind of Goodhart's law fallacy problem. But it's something to just keep an eye out, because I think that subjective idea of what that threshold should be, because 0.025 is kind of like you must be this high to ride type criterion, at least a minimal criterion for most trials. It's going to be a more engaging process to find if this is going to be subjective or changing from trial to trial. More powerful, more flexible, more reflective of reality in many ways, but also more controversial and more difficult, and again the legal stuff and maybe more problematic. But anyway, similar considerations for adaptive interference if you're worried about type 1 error control. They also do go into some of the decision theoretic approaches, like risk-benefit approaches. We've seen some trials. I know there's a good webinar on one of the early GLP-1 trials from Eli Lilly for the kind of generation 1 GLP-1s, not the modern ones that were obviously very popular at the moment. And this is a really interesting area, really wide-ranging, and basically kind of integrating all of the kind of safety signals, the kind of cost components, and kind of integrating all of that together for the sponsor, and then also making sure that it's congruent to what the regulator values as well. Huge area, could have entire webinar series about that by itself if you wanted to. And then obviously they then, as usual, place this big focus on that you should be defining and verifying the operating characteristics of your study. And obviously a lot of these should be to some extent be pre-specified. Basically they say you need to verify across a range of plausible scenarios. So you can't just set the control proportion to 0.5 and then verify the null of 0.05, 0.5. You need to be verifying across a wide range of plausible scenarios to ensure that the type 1 error control or other operating characteristics are being controlled appropriately. If you're doing type 1 error calibration, they argue that you should not only be verifying the type 1 error rate under the null, but also verifying things like the kind of traditional statistical power, the mean square error, the bias, and your kind of same set of criterion as you would for a frequentist trial effectively. I've seen some commentary suggesting that that's a bit excessive and that this is going a bit too far, that type 1 error might be sufficient by itself, and then you can kind of stick to the more Bayesian type approaches for verifying the other operating characteristics, like how likely it's to succeed. But it's an interesting debate. I'm sure it'll happen through the comments on the guidance. And then they say that for a pure Bayesian trial or a direct Bayesian inference trial, they are likely to have their own criteria. So they mention explicitly Bayesian power, the probability of being correct. And basically the idea that you must account for kind of two types of priors. So they mention analysis prior and design prior. So the analysis prior is kind of the one that you're actually going to use in your clinical trial. And so it basically exists to say, like, this is what we're actually going to be doing this, and therefore it is probably your most important prior because this is the one that will actually be used when, you know, actual data is taken from your clinical trial. But they do mention the value of design priors, which is basically a set of priors which might reflect what your belief is, like your more kind of subjective belief or a skeptical scenario or an optimistic scenario. And the idea here is that you can simulate what will happen under these design priors to get a better understanding of the operating characteristics of your trial for a wider variety of situations and what could occur. And that might help you then calibrate what your analysis prior should be. If that's still up to, if that's still in the construction part of the process. And so, you know, evaluating over a wide range of design priors is something that they very much encourage and something that you should certainly keep in mind. And so, sorry, I should make the more technical point that obviously the analysis prior is what's actually being used when you're actually doing like your actual analysis. The design priors are the ones that you're generating data from effectively. So, you know, in the design prior case, you're basically saying from an optimistic scenario, if we generate data from that, the analysis prior is still fixed and the one that's using the analysis, but how do those two interact to create the, you know, probability of success and other type of criteria? So just, yeah, I should make it more clear that that's what the design prior is versus the analysis prior as well. And I would say that this prior distribution consideration is what's given the longest section in the guidance, which is 29 pages, is around 10 of the pages nearly. And basically they argue that prior distributions need to be pre-specified and they need to be done in a systematic and transparent manner. So I think pre-specification is one of the big things here. This is a situation where they expect you to come to them at the point at which you're looking to get a protocol verified to have done the work of constructing a prior appropriately. And then they talk about three types of prior: non-informative, skeptical, and informative. This middle one is probably the least relevant, and I think seen commentary that it mightn't be appropriate to put here, but we'll talk about you know, non-informative, informative primarily here. So non-informative, this is basically just for the idea where there is quote unquote general uncertainty. Basically, we don't know what the efficacy of our treatment is going to be. There's just, you know, not a lot of class of previous drugs. We have minimal evidence from previous stages, whatever the scenario is, and therefore the idea of a non-informative prior is one which will have be quickly overwhelmed by trial data. So basically, you know, once you have a reasonable sample size, the data will effectively contribute a much larger proportion to posterior than the prior itself. And there's concepts such as effective sample size that can be used to illustrate this. So the effective sample size is kind of like how many subjects were used to construct the prior based on how strong it's expected to have in the inference. And therefore, you know, if your ESS is, you know, similar or even larger than your actual expected sample size in the study, that it's, you know, something to be aware of, because that's basically saying we think the prior is really useful, and we're happy to say that even if the data starts disagreeing, that the prior should still have a strong influence on the posterior, on the actual inferences that are made at the end. And so what they argue is that makes a lot of sense. It's a lot less controversial, so it will generally require less justification than an informative prior, but it can be useful when minimal data available, obviously, and it can also be useful if you're trying to calibrate an informative prior in terms of things like the affected sample size, and if you want to do things like sensitivity analysis versus the informative prior. So I would say that, you know, including like a standard kind of non-informative prior in your model is good to have as part of the sensitivity analysis, just to say, okay, well, if we had gone non-informative, here's what would have happened. And obviously we can see that, you know, we lose a lot of the value of doing a prior in the first place by not choosing to use informative prior when the informative prior had data that was, you know, valuable and useful in this particular context. That's basically the argument that you're kind of making in many cases. And they do know that you still need to verify the operating characteristics of a model that's using a non-informative prior, because there are certain circumstances where you might see implausible values occur, or under certain transformations, a non-informative prior can turn into an informative prior. So you need to be careful about that. I think one commentary by Kurt Veal of Berry Consultants kind of talks about a kind of toy example where you have like a beta epsilon epsilon prior for a proportion. Basically just argues like if you have like a 5 subject trial and you ended up with 0 successes or 5 successes, the extreme case, that epsilon epsilon, which is basically just epsilon's an arbitrarily small value, so we end up getting kind of a U-shaped prior, where most of the distribution density is at the extremes of 0 and 1. Obviously the beta is a 0 to 1 distribution, and basically if it's 0, all the density ends up at 0, and if it's 5, all the density ends up at 1. And you would argue that maybe that's not the behavior that you would like to see. That's kind of been a little bit like that's the prior kind of doing a lot of the work, as it were, in that particular case. So maybe something to keep in mind there. And obviously, just something that experienced practitioners would be aware of, but something to keep an eye on, especially if you're someone who's looking to get into the space without experience previously. Skeptical prior is given some brief mention, saying it's maybe appropriate if self-similar treatments have failed, or for type 1 error calibration purposes, say it was a design prior, or might have some use in adaptive designs. I would say that, there's not many sponsors that will deliberately go out of their way to say our drug doesn't work. So I'm not sure how much the skeptical prior would be used in practice. Maybe we'd be better kind of discuss this kind of part of the design prior concept rather than kind of say at this stage, which was kind of maybe more talking about the analysis prior. So some debate about that there. And then informative priors, and these are basically the kind of biggest section here in the priors discussion, and justifiably so. And basically they argue that, if you're going to be using an informative prior, you need to have a justification for using an informative prior in the first place. So this is quite easy if you're talking about current examples, if you're talking about, say, rare diseases or pediatrics, where obviously, rare disease, we don't have a huge number of subjects by definition, and therefore being able to share from previous studies which have tried drugs in the similar space on the same disease or on similar populations or similar diseases. This is where, an informative prior would make a lot of sense. And obviously, you just need to make sure that the implementation has alignment. Basically, the process ensures alignment between the sponsor and the FDA. Like, you don't want a situation where the sponsor hates the prior and the FDA loves it, or vice versa. You want one that kind of creates alignment between everyone, and that everyone agrees that this is a reasonable snapshot of what the actual evidence is for what we expect to happen with our new therapeutic. And obviously, there's necessarily a bit of positive bias if you're doing all this work to get a drug out there for various financial, ethical, and just personal reasons. And so you just need to be, keep yourself a little bit skeptical in the sense of not getting too ahead of yourself when doing this process. They make some notes just at the start saying account for the prior. You should be thinking about the idea of a prior data conflict, say exploring via sensitivity analysis, and obviously thinking about the fact that prior construction and the usage of the prior should be a multidisciplinary project. This isn't just something for biostatisticians, it's something which should hopefully create a really useful arena for all of the different stakeholders, for the physicians and the patients and the other scientific experts and the biostatisticians and the regulators to come together, and the sponsor to come together and to think about, well, what do we actually think is going to happen, or what do we think is realistically going to happen, and using the information to discuss, the actual contours of the trials. I think every statistician wishes that there were more opportunities to discuss, what's the objective of this trial, what should we be doing, why are we using this endpoint, and stuff like that. And this kind of prior discussion about the prior is one arena in which that can actively come, and which the scientists and the domain experts can then get across what they believe or why they believe it and all the evidence they can bring forward to it. And they basically then, the FDA provide a kind of process here where the first step is to identify and review all the available evidence, and there should be, set out initially like a pre-specified plan of, okay, here's all the evidence sources we're going to do, or all the types of evidence we're going to consider, and then how we're going to merge those two together, as we'll talk about in a second. So this could be trial data, this could be PK/PD information, this could be real world data, this could be expert opinion. It's not used in this context, but later on they talk about the expert elicitation and stuff like that, which is an interesting concept itself. We'll talk about that much here. And basically, there should be a pre-specified idea of how we're going to, account for the quality, reliability, and the relevance of each of these sources. So recent, like if you exist in the world where you have patient level data from a recent randomized controlled trial, that's ideal. But of course, that's not really realistic in many situations, to be fair. So, basically, but you do need to have an idea of like, okay, if that's the gold standard, from an RCT, patient level, same estimates, same inclusion exclusion criteria, et cetera, that's ideal, but obviously not something that you're realistically going to have unless you exist within a wider program within, say, a larger biotech. But you should be thinking about, okay, what quality of evidence we'd likely to get, and then how we're going to combine it together. I think they do give some some ideas that, like, a little bit later on, that methods that are used in, say, meta-analysis and similar areas might be good inspiration to think about the kind of methodologies that would make sense to start combining this evidence together. So you said next, you're basically going to construct your prior using some pre-specified and well-documented process, and you need ideally methods and models that account for the relative strength and relevance for each of your pieces of evidence. And they also talk about things like exchangeability, bias, functional dependence, and then a major focus on discounting. So exchangeability is just basically saying that all the trials can be exchanged with each other, because the data, if you exchange the data with each other, it wouldn't make any difference effectively. If you swapped all the labels effectively, it wouldn't make a difference to the inference for the prior construction effectively. There's various different bias. There's certain parameters that might make sense to construct a prior for, but which may not make sense for other parameters. So for example, you may consider this data source to be valuable for calculating the prior for a variance, but not for the effect size. Those kind of things to keep in mind. Obviously, the bias is just something you should be keeping in mind when thinking about the strength and relevance of a particular source of data. So goes through this in a decent amount of detail. And then discounting, which is kind of a terminology which seems to have got some hackles up, because discounting is considered to be kind of a phrase associated with what's called static borrowing or static discounting or discounting. And basically, there's these two ideas. They have static or dynamic. So static basically says, how much the prior will influence the final analysis will be the same no matter what the actual data is, or how much, say, the prior and data disagree with each other. Coming back to the prior data conflict, whereas dynamic basically says that the borrowing is proportional to, most likely, there are other criteria, I suppose, in theory, but most likely in a clinical trial, to the self-similarity of the prior and the data. So effectively, the idea there being that if you have a prior and you're doing a clinical trial and the data agrees well with the prior, then you should use that prior more effectively. Whereas if they're disagreeing a lot, then you should be using it less. And so basically, this is kind of a classic Goldilocks problem, where basically you want a prior that's strong enough that it provides some actual value to your trial. Like, you're not, it's actually going to, if you have good previous data, previous evidence, and you're using totality of evidence, and you combine this with this new evidence that you're getting from the clinical trial, but those combine to create a stronger, more purposeful inference, especially if they agree well, but not a prior that's so strong that if your trial completely disagrees, that suddenly you're in a situation where the prior said it worked, the data says it doesn't work, and the conclusion is that it works because the prior had such a strong influence, like a strong effective sample size, for example. And effectively, I would say from the regulator point of view, they're more worried about that latter problem of the prior being too strong. That's probably what you're going to be concerned about. And I think most practitioners would also be worried about that to a large extent. So you're probably going to err more on the side of maybe a little bit too weak than too strong in many cases, unless there's a good justification for it. But it is a Goldilocks problem, kind of finding that sweet spot where the prior is adding value, getting you basically higher power, lower sample size requirements without creating a situation where the prior is overwhelming the real data that we're getting from the real trial, which obviously is kind of the gold standard evidence compared to most other sources of data. And so, as I say here, just to mention here, that kind of dynamic approach is more commonly called dynamic borrowing in the literature, whereas this dynamic discounting seems to be a new terminology. So just be aware that discounting, if you're reading in the literature, probably means static borrowing, whereas dynamic borrowing is what they mean for this dynamic approach here. Expect maybe there might be some change in terminology there. We'll see if the FDA wants to establish this. I think the discounting idea, discounting as a phrase, makes a lot of sense, because it's basically making the argument explicitly via the terminology that it's about, how much do we want to weigh the prior, how much do we value the prior, basically, or how much do we value the prior versus the actual data that's coming into the trial. But it'd be interesting to see how much they cleave to that effectively. And then they do go through some of the kind of large number of the kind of most probable approaches you'll see used for borrowing / discounting. So you'll see the power prior, which has a static version, but really most commonly these days will be discussed in terms of a dynamic version, where basically the proportion of borrowing is set at some value between 0 and 1. Mixture priors, obviously you might have multiple priors, like a skeptical prior and an optimistic prior, and then basically deciding how much you should borrow from each of these. More commonly would be like a non-informative prior and some kind of informative prior, and saying how much should each of these influence the inference, depending on how much they you agree with the informative prior effectively. Bayesian hierarchical models and elastic priors. So these are all given as example of methods that might be of interest. And then effectively at the end, you should quantify explicitly the effect of the prior via appropriate metrics. And you should be doing this via things like the prior mean, the effective sample size, and this would often be done via things like simulation, via sensitivity analysis, where you're kind of viewing over different design priors as well as that. And then they do say that if you have, after the trial, there should be some post hoc qualification, basically saying, you know, how much information do we end up actually borrowing, because that can be useful for calibration and discussing future trials. And the FDA probably values information in terms of as these emerging norms come around, and then how much the prior and the data conflicted. They do say there's arguments saying that type 1 error itself can be used as a mechanism to talk about the performance of the model. They basically say don't do that. They say it's not a good one. So just a note there. If you've been involved in that. So basically they're saying sensitivity analysis is always very important, simulation always very important, and many of these points are also made in the 2019 adaptive design guidance, which has its own Bayesian section, and then that's also brought forward to the ICHE 20 adaptive guidance as well. Then they make some, and that's kind of the biggest section there, and then there's a few bits at the end. Some of them are longer just because there's a lot of bullet points in the final section about documentation, but basically they make a strong point that the estimate strategy does need to be coherent here, especially ideally between the sources of prior data, prior trials, and your current trial. And they basically say, you know, ideally there is alignment between the prior data study, but they even make the argument that if you have patient level data available, then you should perhaps reanalyze the data which is going to feed into your prior, using the actual estimate strategy from your proposed study. I don't know how practical it is in practice, but for larger biotechs, that might be something they might think about, because obviously they might have a longer running programs that have access to that kind of data. For smaller, medium biotechs, that's maybe less realistic. They mentioned software briefly, which they do in most of these guidances, like in adaptive design. They obviously emphasize that the FDA does not require specific software, but that it must be reliable, as the ICHE 9. And they just mentioned that, you know, most Bayesian inference these days uses approximate sampling algorithms, MCMC type approaches, Gibbs sampling. I'm sure you're aware of the type of phraseologies. And basically just say that given the practicalities of Bayesian analysis, this needs to be accounted for, you know, ideally via simulation, sensitivity analysis, etc., to ensure that the approximation is appropriate and works for your particular trial. And that becomes particularly true if you're doing a more complex model. So the more complex you are, the more you'll need to kind of verify that the algorithm that you're using, JAGs, etc., is appropriate for that particular trial. And then they outline in detail the kind of documentation requirements for the planning stage in your protocol, and then also the reporting stage if you are including a Bayesian analysis. So obviously we're a little bit later here. There were a few weeks past when it was initially published, but the draft guidance feedback is open to March 16th. So there's still a couple of weeks there for you to get your comments in. I would say the commentary that I have seen has generally been highly positive. And they said this is a very detailed guidance that provides practical, useful information to do Bayesian inference, and that in its kind of phraseology and the way it talks about things, it is effectively placing Bayesian inference on an equal footing to frequentist inference in clinical trials, and also then tries to bridge the gap to ensure that these can kind of get along as well going forward. I don't think this is basically saying 0.025 is going away completely, and that there's many scenarios where it's likely to still be something you could expect to be discussed about. But it's basically saying that if you have a strong justification, especially as I said in the context of borrowing, and you have actual real data that can be used to improve your inference, that then the totality of evidence argument is now being weighted a bit higher than the kind of the RCT gold standard argument, if they were like that. And this kind of reminds me of the a joke paragraph, like a graph from Darren Daly saying, like, you know, the RCTs versus everything else is like the well-designed stuff versus the not well-designed stuff is the kind of evidence pyramid. There's a good joke one there if you're interested. And yeah, so basically, I think one thing that's obvious is the amount of engagement that there was from the Bayesian community in the generation of this document. I think Frank Harrell, who's obviously well known in this space, was part of the groups who were involved in the external groups in terms of helping define this guidance. And I believe he was on the In the Interim podcast last year, and was very effusive about how good he thought this document was. Some notes that I think in terms of the negative feedback, which I think there isn't really that much of, there's some notes on some terminology and just also ensuring that the phrasing is open. Basically, there's always concern that traditionally with regulatory documents being quite close-minded, or just saying, okay, we've done this, but we're not sure about this, and that means that sponsors are quite afraid of kind of bringing that forward. This is, I would say the language is open, but there's just some areas where maybe just keep it open, make sure that future usages of Bayesian statistics aren't being hampered by the language, rather just focusing on the ones that are already successful, like borrowing and such. There is a significant overlap between this guidance and certain current priority initiatives from the FDA. such as plausible pathways and continuous trials, which we'll talk about in a second. So that is something that gives it even more heft, I would say, from the top in terms of the regulatory perspective. I think the big and most interesting debate will be about the impact of the emergent standards that comes from as more and more of these trials occur. That's already occurring. Like we have things like the CID program, and we've already had trials, say for during COVID-19, that use Bayesian methodology for the vaccine trials. But what will be really interesting to see is, this kind of, there's almost a tension between obviously the power of the Bayesian approach, which obviously kind of brings some degree, or let's say like a somewhat more greater degree of subjectivity versus the frequentist approach, in some ways, and just in terms of it's like less restrictive. It's more flexible. Like the more flexible you are, the less restrictive you are in some definitional sense. But that also means that the discussion or the back and forth between the sponsor and the regulator about, okay, like what is the threshold, for example, for the posterior probability or the posterior probability success or something like that, and then kind of how that evolves over time, and then like the kind of like if one trial gets this threshold and the next sponsor gets a threshold that's more restrictive, then what kind of debate will that create, and like how will the FDA and other regulators deal with that degree of subjectivity, I suppose, and that does create a concern. And then that could even escalate the legal stuff, that the FDA has strong broad powers to not have to deal with that, but it could come up eventually. And so it's just a really interesting, like, just keep an eye over the next few years as Bayesian trials become more popular, which I'm almost certain that they will. Like, what are the emergent norms that come from this? Because I think, the people who have been doing Bayesian trials right now have been trendsetters, people who have, a lot of inbuilt expertise, who deal with, consulting companies which have great expertise in Bayesian analysis. But as more and more people do this, and basically, they try their hand at it, for lack of a better term, especially in earlier clinical trials where, there's kind of other priorities rather than just like incredibly high standards for the evidence generated from the trial itself. What's going to come up from that? So something to keep an eye on. And I think it's already very clear from the people who already are very influential in Bayesian clinical trials that they don't want to see this as a kind of Trojan horse for reducing evidentiary standards, or at least unjustified reductions in evidentiary standards. So I think all of them would say, like, there's definitely things we could do. Like, we could go from the two trial to one trial thing, or including previous data. There's ways you could do that badly, just like you can do frequentist statistics badly. But what they don't want to see is that this all gets tied up into a larger, broader move where, treatments that don't work end up being approved, or treatments that are harmful get approved more often than they did right now. And I think a huge amount of push from the innovation side, not just to make trials better at getting drugs approved, but also making them better at picking out the ones that are bad or which are unsuccessful earlier as well. So it's like, you won't find a statistician who won't say that there should be more futility analysis in sequential trials, for example. Just a note here that obviously the FDA is kind of the torchbearer here, but the EMA methodology working group just published a couple of weeks ago itself a concept paper on Bayesian analysis. They discussed the lack of clarity, and it goes through a long list of all the things that could be discussed if you're talking about a Bayesian guidance. But they are aiming to have a reflection paper to be finalized by 2028, so a draft version probably next year. So don't be surprised if I have Bayesian stuff in here again this time next year. I just keep an eye on that. I think the EMA is generally considered to be a little bit more skeptical than the FDA traditionally on these type of issues, like innovative design and stuff. But they did have a big meeting on Bayesian methodology last year, which I think I forgot to put in the slides. But if you're interested, let me know, and I'll send it on to you, the URL for that meeting there. So it is. I think what you're going to see is, if this is successful, other regulators will kind of push into this and use this as well, is the most likely outcome. And then the big question then is, okay, an ICH Bayesian guidance, that's obviously several years in the future. And then the question is that it's like a separate guidance, or, does it make sense to just make it another addendum to ICH E9, which is about the statistical issues in clinical trials, similar to how the estimate strategy was dealt with. Or maybe the Bayesian guidance just needs to be, something that's inserted more, like it is mentioned in various parts of the existing guidances, but made more like fleshing out those parts and making it more thorough, effectively, assuming that there is a regulatory alignment on that on the global level. Okay, so the Bayesian stuff is obviously kind of really exciting at the moment, and obviously as a biostatistician, it's kind of what catches my eye, but there are lots of other things going on. And so part two, just go for continuous trials. So continuous trials is this general concept that basically we want to supplant the traditional 3-phase trial structure. You can even argue it's like 4-stage, depending on like if you have phase 2A and 2B, but we'll get into that. Obviously, what we want is to have these kind of large, fluid trials where basically the data is being shared continuously between the sponsor and the regulator. Basically, then you could have parallel meetings or early meetings. Basically, the whole idea is that the regulator always knows what's going on or knows a lot more what's going on at each time. And then it also makes it much easier to add in additional treatments or changing the centers. And then all these processes become much more seamless, effectively. And obviously, this is paired with multiple other initiatives to decrease review timelines, reduce administrative burden, and trying to streamline everything. And I would say like this is a direct reaction to obviously a major shift that's occurred in the last 5 years in particular in the where clinical trials are actually being run. So China in particular has seen substantial growth. It's now, I believe, it depends on what you read, but I know certainly in early stage clinical development, China is now the largest region in the world, and that continues to grow. And you're seeing major biotechs, if you read the pharma news, who are licensing drugs from Chinese companies now. And basically, the argument is that China has massively streamlined the administrative side of, in particular, of the start ramping up the clinical trial process at the early stage, and that's also having filtering into the later stages. But the early stage in particular, there's a big focus on in terms of the administrative side. And there's just things like the assumption. So rather than assuming like a skeptical approach, there's an assumed yes. So unless someone objects or the regulator objects, it goes ahead. It's kind of one of the kind of normative changes that they've made that seems to be very very encouraging to biotech, biopharm sponsors. Now, in reality, the idea of like continuous or seamless trials has been, something that's been ongoing for a while. There's been various initiatives. So in earlier stage trials, you've seen Project Optimus, which is kind of redefining how phase 1 particular trials should be undertaken. And that's not just response to a need for more efficiency, but also a desire to have, especially in oncology, better models for immunotherapies and modern oncology treatments compared to cytotoxic treatments. CID has loads of case studies for early and late stage trials. We discussed that briefly previously and in previous webinars in previous years. And then recent regulatory guidances on adaptive design, which we've referenced previously, on decentralized clinical trials, on real world evidence, etc., platform trials, et cetera, master protocols. There's a whole bucket we've kind of briefly mentioned at various points at this point. And then basically the kind of one big thing that's happened this year, and there's a bit of controversy about this because I believe it was cited as one of the things that there was a negative reaction from Scott Pasdor, who recently left as the chief director of the Oncology Center for Excellence under, I won't get into the politics of it, but under interesting circumstances. But basically the 2-trial rule has now been officially got rid of. Now, if you look at the statistics, I think just over 50% of all of innovative, like all drug applications that were approved last year were already 1-trial rules, and we've seen the emergence of multi-regional clinical trials, which have large large sample sizes, and obviously the regional the global aspect of them gives you lots of information that can then be used to kind of show, okay, this drug actually does work. So it's kind of going out the door anyway, but to explicitly basically say we're moving from a default. The default assumption is it should be a 1 trial, one large, defined trial over to, like, I suppose in theory, smaller trials. So it's an interesting change there, and obviously that makes it more compatible with things like multi-regional clinical trials and other things which could help accelerate clinical trial development. And so from a biostatistician point of view or clinical trial design point of view, this obviously it's not the same word, obviously, but it kind of fits nicely, which is this idea of seamless trial methodology. And obviously, just to mention that there's kind of 2 types of seamless trial. There's operationally seamless and there's inferentially seamless. Obviously, as a biostatistician, and we're going to focus a bit more on the latter today, but just to say pretty much every inferentially seamless design is going to be an operationally seamless design. I'm sure one could contrive a way that doesn't happen, but for practical purposes, we consider that to be true. And these designs have been rapidly evolving in recent years, and this push to continuous trials seems like an optimal time to kick in there and make this even more prevalent in the clinical trial space. But there is this big question of where the reality between this kind of lofty idea of having a trial that just goes on forever, or just kind of happens, everything happens, people go into phase 1, they get to what's equivalent to phase 3, and it's just the same trial. You get to share all these resources, share the infrastructure, share the patients, share the statistical methodology. We'll kind of see where things fall between that idea, our hope, and the reality. of what actually happens in clinical trials. So just to give you a brief overview, I suppose, of what we're talking about here, in terms of like a sequential clinical trial, we have preclinical, and we have phase 4 post-marketing, which we won't get into today. But basically, you have a phase 1 trial, where basically usually what we call a dose escalation trial, especially in oncology. We're usually basically trying to find the maximum tolerated dose. We're trying to find the safety profile of this drug. So this is your first in human trials. This may be on patients who have the disease of interest, but it may not be. It may just be on healthy volunteers. And more recently, there's been a bigger focus that maybe we actually want to find some measure of the actual biological effect of the drug in terms of the positive sense. So that might be PK/PD type information. It might be some kind of response, tumor response, something like that. Something hopefully you can quickly measure, as it were. And then phase 2 is basically the first stage, usually where we're trying to establish not just the safety, but also the efficacy. So we're usually looking about the proof of concept, which is the kind of 2A, and then also the dose finding, which is 2B. So dose escalation is usually, you're talking about phase 1, dose finding usually phase 2, though you'll see very often that you can see, especially dose finding might be used for phase 1 as well. But anyway, these two stages, sometimes which are merged together. So for example, there's the NC-mod methodology, which can combine these kind of two inferentially together. But effectively, the idea here is to first prove that there's a sufficient signal that the drug actually has the response that we expect. So you might have some surrogate endpoint, like overall response rate, that you might use, or maybe progression-free survival if it's very difficult to get a good surrogate. And basically, we just want to establish, okay, there's some evidence that this drug actually does what we think it does, for example. And this is the stage which most trials fail, that are going to fail. Like there's a very high attrition rate at this stage. And then we get to phase 3, which is our pivotal trial, our kind of classic confirmatory trial, where we want to establish in a large randomized trial that the drug is both effective and safe. And that's kind of the classic ones that we're all familiar with, which are kind of the biggest expense, but which have the highest degree of regulatory oversight as well. And I suppose if you're thinking about the current widely used methodologies used in quote unquote seamless trials, there might be basically a transition from to this type of approach, which is basically phase 1, 2, and then phase 2, 3. And there are ways you could potentially link those together, but that's kind of the classic thing. So basically what we do is we have one phase where we're combining the safety, ensuring the safety of the therapeutic, and then also trying to find the candidate doses that make sense for further evaluation, both on their safety profile, but also on their efficacy profile. So we still have dose escalation as being kind of the starting point for this, and probably for safety as well. But basically there will be ideally some integrated approach, which we're also balancing that against the doses which we think have acceptable toxicity, but also the best biological activity, the best efficacy, or what we expect to be best efficacy. And then basically, our dose escalation then might transition seamlessly into a design where we add additional subjects to the set of doses which we think are useful for characterizing, say, the dose response curve, or which we just think are the most likely to be brought forward directly into the next stage of the trial. So this would be something, say, cohort expansion type trials and variants thereof. And I think more recently, the FDA, in terms of Project Optimus, has kind of suggested that even randomized dose response things should be kind of seamlessly interwoven into the dose escalation part, or potentially that could happen as well. And then basically, okay, so phase 1, phase 2 basically gives you either explicitly just a set of doses, or hopefully a dose escalation curve, and then that can be then used to inform, okay, we have a candidate set of dose levels that we think would be valuable to assess for further for efficacy on, say, a more on a endpoint which maybe requires more time to get to fruition, so PFS, some kind of response rate, and then effectively we're going to evaluate the dose levels that we select, and then the best performing dose, or maybe 1 or 2 dose levels that perform best, will then be brought forward to the confirmatory stage, and that'll be done seamlessly, so that all the patients that were in the phase 2 trial on the control arm, and then on the arms that are selected from the treatment group, arms are selected, well then those patients are included, both the phase 2 and phase 3, with some kind of statistical methodology used to adjust for any type 1 error inflation, for example. And in theory, one could go from phase 1, 2 directly to phase 3, and that does occur in some situations if there's sufficiently good evidence, or if the sponsor wants to take some degree of gamble, as it were. But I think, this is kind of what I would say is kind of maybe based on what we're doing right now, the kind of idea of what a seamless trial might look like. But then, of course, the idea of like a continuous trial, and obviously this is kind of a fairly vague concept of right now. We haven't seen much flesh in the bones yet, and it interacts with a whole bunch of stuff. It's already been undertaken. So how much is it going to be just building on what's been done already, or what's being suggested already, and how much is a completely new paradigm? But the basic idea here for, I suppose, a fully continuous trial would be that you'll be doing dose escalation. You would then end up with something like a, say, cohort expansion for further evaluation of the doses that have the best acceptable safety, but also the best chance of efficacy. And then those are brought forward seamlessly to some further evaluation, maybe on the same endpoint, maybe on a new endpoint. And some correlation can be characterized within that. That's brought into phase 2. Did you do the dose finding? You're basically evaluating which dose or doses are best, and then whichever dose or doses win and are brought forward to phase 3. Those are then entered into into the confirmatory stage, and then obviously you get your approval based on that. So there are scenarios, like I think if you're talking about things like using basket trials, and if you're seeing kind of not really used for new drugs, but for, say, additional off-label, like kind of getting approval for additional indications for a drug, you're seeing things like platform trials, basket trials, these kind of massive protocol trials, which kind of fit into this candidature. But for like a new therapeutic to go from phase first in human to approval with having a operationally seamless, and then probably very difficult to imagine how to be inferentially seamless, but potentially even inferentially seamless design. That is kind of, I suppose, the platonic ideal of what this idea is, right? And I'll be interested to see how that could work. And I do not claim that this is like an accurate idea of what this is, but this is kind of if you were to ask me, just jot down a truly fully continuous trial. I suppose this is kind of my idea of what that might look like effectively. But this is speculative, so please don't hold me to this if this ever comes to fruition, or if it's very different from the continuous trials documentation or stuff that we end up getting. I suppose I provided here just some brief overview of what a seamless phase 1, phase 2, and phase 2, phase 3 trial looks like. So seamless phase 1, phase 2, we're combining dose escalation with some degree of the proof of concept dose selection from phase 2. Usually, ideally, hopefully some level of the dose selection, because that will inform the quote unquote proper dose selection, dose finding trial that's kind of phase 2B, whether that ends up being a phase 2B3 or just a phase 2B standard one. That's left up to the sponsor. So dose escalation, as I mentioned, is focused on the safety signal in first in human trials. And basically, it's traditionally focused in oncology, in particular on the MTD, the maximum tolerated dose. But you can see here, like the FDA talks about the fact that these targeted therapies, these immunotherapies, have a very different type of response curve for toxicity and efficacy than traditional cytotoxics. So, in cytotoxics, you expect them just to be monotonically increasing, whereas for efficacy in these targeted therapies, it might just taper off. And so this kind of would argue then that, okay, we don't want the maximum dose. We want the one that has, the kind of the dose that is the plateau dose, basically, where it starts plateauing, and it's not really worth increasing the toxicity in the trade-off for that purpose at that point. And so this is kind of a response to this problem, but then it kind of feeds into many of the other stuff we're talking about here about seamless trials in general, in terms of these more modern dose escalation methodologies. So it's a very actively evolving area at the moment. So it's kind of hard to say exactly what the best way to do things is, but it's very interesting. And so a seamless trial would ideally have some mechanism to basically balance this efficacy and toxicity considerations together, and then basically find the doses that are best, and then potentially do, say, a cohort expansion, where effectively that cohort and that dose will be given, an additional cohort will be given the dose or doses that think are worthy of further evaluation, and maybe even a randomized comparison, like the most recent guidances from Project Optimus kind of suggests randomized assignment between doses, but that's kind of up in the air, as it were, at the moment. And basically, you can see certain strategies already out there, such as the FTOX design. The Yan paper is really talking about the FTOX design, which is kind of an older design. But basically, kind of here, kind of a cycle of, okay, what's the decisions we make to ensure that we kind of pick the best dose that balances the efficacy and futility, the efficacy and safety criterions. And as I said, there's reasonably comprehensive FDA guidance on expansion cohorts provided here, and I said there's one about the kind of maybe the more modern approach to dose finding, which is kind of more of a hybrid between phase 1 and phase 2 considerations. It's mostly focused on phase 1, I would say, in terms of terminology, but it's kind of arguing, I would say, implicitly for some level of seamlessness between phase 1 and phase 2. It's not the direct point of that guidance, but I think it argues that this could be it's kind of leading things in that direction, I would say. And then we have seamless phase 2, phase 3 trials, and these are kind of in a similar bucket to multi-arm, multi-stage designs, or like platform trials and umbrella trials. But effectively, what we're doing here is we're trying to, we have a set of candidate doses. We want to pick the winner doses, basically the ones that we believe work the best. And obviously, usually there'll be a criterion saying that if all the doses suck, then we just stop the trial early. So you still want to have that kind of futility criterion that's very common, early futility criterion that's common in phase 2 clinical trials. And you may have, futility assessments within the phase 2 trial itself, and then a final futility criterion before the no-go decision from phase 2 to phase 3. So those, there could be multiple futility assessments if you're interested in that. And effectively, all the data from the control arm, which obviously will be expected to be, will be in both parts of the seamless phase 2, phase 3 trial, and then obviously the dose or doses that are brought forward. All the data from those subjects in phase 2 is then brought forward into phase 3. And I think there's been some interesting work on this. I think there's a work from people involved in Berry Consultants that said that the phase 2 trials, seamless phase 2, phase 3 should perhaps be bigger than they usually are. I think there's the natural inclination that's kind of like the phase 3 trials are the big trials, and phase 2 are the small trials. So we just take like a natural phase 2 trial, which is like maybe 100 or like a 50 to 100 or like 100 to 200, something like that. And a phase 3 trial is like hundreds or thousands. If we just put the traditional sizes together, you don't really get the efficiency you would expect from a seamless approach. And so that maybe if you're willing to invest a bit more into your phase 2, given that data ends up coming forward, you'll end up out with an overall trial structure that has higher power and less sample size requirements. So you could trade those things off against each other if you need. And therefore, maybe larger phase 2, the larger phase 2 part, the dose finding part, the traditional phase 2 would make sense if you're going to do this seamless type design. There's also lots of other papers that go through the same analysis and come to a similar conclusion. But obviously, in the moment, how compatible that is with the actual, say, financial model that you have for your particular candidate therapeutic is a different question. Obviously, you need a statistical analysis that can account for the kind of multi-arm, multi-stage design, combination tests, group sequential MAMs, Bayesian methodologies are available. And these are all kind of covered within adaptive design guidance. So ICH E20, the FDA guidance, the EU guidances. There's obviously some overlap with the platform trial guidances that if you're looking to go a bit more exciting or more interesting there. Treatment selection basically is the main section you'd be interested here. But technically, maybe response adaptive randomization might be of interest as well if you want to get involved a bit more excited there. And then I suppose in terms of challenges to this continuous trial thing, because it's not going to be easy, nothing worth doing where it is. So speed may not always be the highest priority for some sponsors. And obviously moving as fast as possible comes with its own set of risks. So, safety considerations, especially in these early stage trials, might be a concern. So I think there's already been evidence and people who've cautioned that, in these dose escalation trials, in these cohort expansion trials, that because they want to get as quickly to the cohort expansion as quickly as possible, that's led to some subjects being exposed to, dangerous dose levels that weren't known at the point. Obviously, you're talking here about cohorts that are usually of size 3. We didn't have enough evidence to suggest it's actually safe. Maybe there was only one DLT in that group, and we kind of said, okay, let's do a cohort expansion because the biological response is best there. And then suddenly a whole bunch of people are exposed to a dangerous dose level. So that's just an intrinsic consideration and something you need to balance across each other when thinking about early stage trials, seamless trials in particular. I think for phase 2 to phase 3, there are some. You want to design this well to make sure you get the value of it. I think in particular, if you're using some of the more aggressive, more conservative statistical methodologies, such as like certain kinds of combination tests, you may end up losing a lot of the value of doing a seamless trial. But I think the biggest concern here is that obviously phase 2, because that's where most trials fail, from a funding perspective, is often, for larger, smaller biotechs in particular, like you need to get past phase 2 to get the next tranche of money or the acquisition that actually makes the phase 3 trial happen. So how much are you willing to invest up front in your phase 2? Are you willing to go for a larger phase 2 in a seamless phase 2, phase 3 program? even though you know there's a high chance of failure, because that's what happens at phase 2, relatively speaking. Does that then mean that you want to do a better proof of concept trial to ensure that you're not wasting more money and quote unquote wasting more money? Now, obviously, in the context of larger biotechs, if you take a portfolio approach, then knowing information about which therapeutics don't work is actually a good thing and should be encouraged. And there's a whole bunch of literature about that. But there's definitely a consideration there in the practicalities of whether a seamless trial makes sense for a particular sponsor. Like the value of these, I think usually the value you're talking about like maybe 20% reduction in sample size. You might consider, okay, like these additional risks and obviously additional kind of infrastructural requirements for a seamless trial versus a non-seamless trial. Is that payoff going to be worth it? Is also to keep in mind. And then obviously just widely here, and this to a lesser extent with the Bayesian stuff, but is this push for continuous trials likely to be a race to the bottom? Because I said this is kind of in the context of like global competition in terms of clinical trials, or just a bonfire of excessive red tape. Have we gone too far in the direction of conservatism? And I think we would definitely argue that the last 10 years is all about kind of peeling back bit by bit the kind of ultra conservatism that might have usually characterized regulatory perspective on clinical trials. But are we going too fast in the other direction? So that's kind of a wider debate about this thing, because I think continuous trials is kind of a concept that's going to bring in a whole bunch of other stuff that is not really to do with biostatistics or clinical trial design specifically, but to do with the kind of infrastructure and the way that regulators and sponsors interact with each other. A lot of the benefit of this idea will probably come from better infrastructure, better technology infrastructure for how data is shared between the regulators and the sponsors. This idea of being able to quickly move data back and forth seems like overall a major positive, and that basically makes it more possible to not just do these kind of strategies such as like seamless designs, but also to do things like parallel things, so where basically the different stages of the trial overlap with each other, but they're not explicitly tied in with each other operationally or inferentially. And so continuous data sharing I think is generally a positive, but it's obviously a major investment required to ensure that the infrastructure is there for both the regulator and the sponsors to get it there. And the sponsors will want to see evidence that they're going to see faster timelines. I know the PDUFA negotiations are happening right now, for example, to be willing to buy into something like this. This requires a major investment from both them and from the regulator itself. And obviously, there's been cutbacks in some of the regulators in the last year, which make that maybe difficult to imagine whether it may or may not happen. As well. So just something to keep in mind there. Obviously, this the faster you go, the better you need to be able to basically get the the therapeutic actually needs to get to patients in this situation. So you need to be better at bringing new sites on board. You need to be better at bringing things like manufacturing to subjects. You may also want to do things like continuous monitoring. So there's like guidances on decentralized clinical trials, for example, that'd be worth keeping in mind. So the idea here is that if you're going to move fast, then you also need to be moving faster all the intermediary stages of getting taking a clinical trial from something that's on a page on a protocol to something that actually happens where treatments are getting into the hands of patients, and then evaluating that evidence for the efficacy and safety of the drug of therapeutic interest. And obviously there's some interaction with other trends such as the accelerated approvals. I said the removal of the 2-trial rule default. And I think one small thing I would say here, I'm not going to make a big point of it, but I think there's been there was a big regulatory change for the FDA in particular, which basically makes it much easier for it to get withdraw approval, conditional approvals in particular, accelerated approvals. And I think that power is probably going to be used more and more. And it's already created some back and forths, I feel like, between sponsors. I think the FDA is generally be winning those back and forths given its new powers, legislative powers, or statutory powers, I should say. But phase 4, traditionally, I did a webinar on this, I think either last year or the year before, and it's like traditionally it was kind of hard to get sponsors to do the phase 4 trials. Basically, like they kind of say, oh yeah, we'll definitely do another trial to evaluate that the treatment has the thing that we said, or we'll get the overall survival after getting approval of PFS. But I think tightening up those requirements, making sure that they follow through, is probably going to continue to be a big emphasis going forward. And you might see more of these treatments getting conditional approval, accelerated approval, which kind of tied to this idea of seamless continuous trials. They might also get more approvals withdrawn when the early evidence doesn't end up stacking up with the actual reality. And then just briefly some other trends. I don't think I'm going to have time for questions. I apologize for that. Just some other things to keep an eye on. Rare disease pediatrics. This is been a huge emphasis put on this over the last 5 years or so, I'd say, basically, especially post-COVID. And you've seen initiatives happening under previous administrations in the FDA, like the ARC program, which includes the Leader 3D educational program, which has some nice YouTube videos and various types of clinical trial questions if you're interested in that. There was like a rare disease guidance published in 2023, a disease innovation hub that was announced in 2024. I'm not sure what that is at this point. That might be something that's going to be amalgamated into a different initiative, but it was announced. And then the huge thing and a big focus of the current FDA commissioner is this plausible mechanism pathway guidance for certain like N of 1 type treatments, like involving genetic treatments and stuff like that. And this guidance was published literally this week, so I haven't had time to review it in detail, I'm going to be honest. But basically, this is basically saying that treatments that could obviously involve gene editing, that these could, be obviously, these are hyper specific to a single subject, but basically the idea being that rather than having to run a new clinical trial for this thing, if you can establish that it works for one patient, for one patient, that the skip and the jump to it working for the next patient may not require like another trial or something like that. And basically just trying to create a pathway here where if there's a very strong biological pathway that can be established, which might be itself have to be established initially in a small clinical trial, like based on some of this guidance about rare disease trials generally, but then that, the extending that forward, even if it involves something that's like a little bit different, or obviously you've changed the kind of the nucleotide sequences and stuff like that, doesn't require kind of going through the whole approval pathway again. So I think a big question is, there's been a lot of interest in this, like how quickly will sponsors adopt this plausible mechanism pathway, like what evolving precedents are we going to see in response to that, and just the general change in views on rare disease. So like Bayesian guidance has a huge influence on this if you're actually doing like a trial in rare disease, in terms of like using borrowing from previous trials, from other subgroups, from going from adult to pediatrics. And then I think a big thing to watch is just like, okay, like if is this something where other regulators suddenly adopt this pathway? Because if this ends up being really successful and leading to lots of relatively quite fast approvals, then obviously some of this kind of global competition stuff I've mentioned might lead to other regulators quickly adopting this approach to gene editing, similar type of treatments as well for rare disease and pediatrics. AI/ML, I kind of talked this a lot in 2025 trends webinar, so I won't go over too much for today, but there's an EMA reflection paper over the whole product lifecycle, which kind of has some aspects related to clinical trial. The draft FDA guidance was published in early 2025, which I think I predicted like in last year's webinar. And there was obviously a bunch of papers I talked about there. Don't have time to talk about today, but it was generally quite positive and kind of very bullish, I would say, on AI usage in the use of AI in clinical trials. I would say the aspects are probably not as directly involved in kind of clinical trial design type aspects. It's kind of more various aspects that kind of move beyond that, including things which might be relevant for the Bayesian stuff in terms of like prior construction, stuff like that. There was a qualification of an AI drug development tool for MASH evaluation 2025, and then the EMA and FDA published a guiding principles document in 2026. All of these are in the references if you're interested in reading these more. I won't have time to go through them in much detail today. Obviously, the finalized FDA guidance I would expect to potentially come out this year after being in draft review for about a year now. AI-driven versus AI-assisted clinical trial design. So I think AI assistance is already quite common, I would say, in many scenarios. People are probably already using it to some extent in protocol development. But I suppose the holy grail would be, this thing does nearly all the work, or it does, and especially starts to make more and more automated decision making. Obviously, in the AI space, huge focus on the agentic framework and how much could that influence our process here. Probably slower in clinical trials because obviously due to the regulatory regulated space nature, but certainly there's a lot of excitement in that in other areas, so expect that to have some influence. I think if you're on AI discovery, which is kind of a separate topic, but obviously we're now starting to see how well do these AI discovery platforms compete against the traditional approach to drug discovery, and then continued evaluation as a clinical trial design tool / assistant. I've seen a couple of papers in the last couple of years talking about it for the usage of things like sample size calculations, covariate assessments, inclusion exclusion criterion, set it like and evaluating site compatibility and stuff like that. So a lot of interesting stuff happening there. And then obviously the big step is in operationalizing that going forward. And then more CT. This is probably my most speculative one, I would say. But basically, there's been a long running guide in terms of like, okay, guidance for multi-regional trials. Obviously, we're kind of focused a little bit recently on the sample size aspects. My colleague Dennis Desmond did a webinar on that last year, and we added some sample size tools based on these kind of proportional allocation and the relative effect size, the regional specific effect size kind of criterion. But I did note in, so this should be 2024 guidance, not 2014, but I did note that the FDA OCE a couple of years ago put out a guidance basically saying, noting a decreasing proportion of US participants. And I've obviously referenced that in generally in other topics today. And obviously, as I said, the two trial rule has been ended as well, which makes it more compatible to like a large MRCT might make more sense if you don't have to worry about the two trial rule in the US jurisdiction. So I suppose the big question here is like, are we going to see from the kind of traditional clinical trial areas like the US EU, like greater scrutiny? Obviously, there have been trials which have been rejected because they didn't have any US participants or minimal US participants already. That's not something that's unprecedented. But are you going to see increased scrutiny of that as more and more of clinical trial development occurs in other jurisdictions? Now, the way it seems to be happening right now is that MRCTs are mostly being done by larger biotechs by licensing Chinese candidates. So that kind of gets around that to a certain extent. But I assume it's quite likely that the Chinese biotechs want to get into that space themselves eventually, rather than relying on licensing, or potentially, I suppose that's one thing they could be, and that might end up, if they start doing that, well, then where will things end up falling? And whether this OCE guidance that included that note ends up coming out this year, obviously with the change in OCE leadership, which I referenced briefly earlier, that's a bit more in the air at the moment. So that's just some other things to keep an eye on, and things that we might end up discussing more in future years as we do these trends things each year. And this is just a review of some of these areas as well. This is actually from last year's webinar, if you're interested in a kind of high-level overview of each. In terms of discussion conclusions, clinical trial innovation continues at a rapid pace in response to multiple challenges to conducting efficient and comprehensive trials, and I would say also in response to different challenges there. Adaptive design continues to grow, Bayesian methods continue to grow, initiatives to make trials more flexible continue to grow as well. Okay, just to mention that if you want to do a demo of nQuery, there's a URL here that will allow you to try nQuery within your browser. You just have to provide your email, you provide a link, and you can just try it within your browser, so no credit card, etc. required. And if you want to get in contact, you go to info@statsolves.com. More info also on the website, statsolves.com. Okay, as mentioned, I've run over time a little bit, so I apologize for that. But I won't have time to go through questions. But if you do have questions, you can email these here. And any of the questions that have come in today, and I see a couple, I will answer those via email afterwards. So I'll leave this. I'll give you a few seconds here to put in any final questions that you have, and then we'll finish up today. So just finally, I want to say thank you so much for attending. I hope there was some useful information in there. And I said lots of exciting things to look forward to in 2026. So I'm very excited to go through those as we see them alongside you this year.